All right, everyone. I was just informed that my most recent manuscript was accepted for publication as of today. So, yeah, this is awesome. So, this is some work for my PhD where we looked at how proteins change in response to simulated microgravity. So, if you want to talk about proteins or you want to talk about microgravity, here's your video. So, this is awesome. The idea is that a microgravity environment is void of external pressures, right? There's no gravity. And if you think about it, biology is quite intertwined with gravity. At least to what we know of, there's no life that's grown anywhere outside of a gravitational force, maybe. In any case, plants will grow up because they're pushing against gravity. Right? These plant cells and our human cells and bacteria and all sorts of life have these mechanosensors where they're sensing the pressure, in this case gravity, and then communicating that pressure to the inside of the cell, and the cell is responding accordingly to all that pressure. And one way to think about this is it's kind of like a production line where a signal then has a step followed by another step followed by another step, almost if you were in a car manufacturing or a beer bottling facility, right? You've got step one, step two, step three, step four, step five, and so on. But inside the cell, you have multiple pathways that are integrated, such that when we consume food or have some stimuli, the idea is that we can break down those molecules and then fit them into one of those pathways and then use them in a multitude of different pathways. And this allows us to have a wide range of diet or different types of exercises, right? You've got the strongman, the real heavy, you've got the physique, you've got CrossFit, you've got this primal. There's all these different types of ways of moving your body, all the way to Tai Chi, Pilates, yoga. And, and in essence, all of these movements, just walking, is communicating to your cells through the same general pathway of this mechanotransduction pathway. So as you get one pathway going and then you add on these other pathways via heavy weights or cardio intensity, or this slow movement of like Pilates or holding a pose, this isometric, then you're adding into these different pathways and changing the way the cell responds. And I love the idea of exercise, right? So studying microgravity for 6 years in my PhD was epic because it is, in essence, the opposite of exercise. It's pure stagnation. So the way we did this was using a simulated microgravity instrument. This was designed by NASA, and I worked with a few engineers to make a larger 1-liter vessel. And the idea is we had a 1-liter control and then a 1-liter simulated microgravity vessel. And then we repeated the experiment 6 times. So the value of having some statistics, usually scientists will repeat something 3 times. In this case, we had 6 times. So I had 6 growth conditions of normal gravity and 6 growth conditions of a simulated microgravity. And the organism I used was a model organism called Saccharomyces cerevisiae, also known as baker's yeast. And the beauty is that this single cell eukaryotic organism is very similar to human cells, oddly enough. There's actually been multiple Nobel Prizes where they used Saccharomyces as a model organism to understand some biochemical pathway that was then applied to human cells and saw that there was a congruency or consistency in how the cell responds at that chemical level. So we grew this yeast in both conditions, and I wanted to quantitate how proteins changed. To do this, I used a technique called SILAC. In essence, proteins are made out of amino acids. So when I added amino acids into the culture for the yeast to turn them into proteins, all of the lysines had an additional neutron at each carbon and nitrogen. As a reminder, neutrons have a mass, but they don't have a charge. So this means that the lysines are heavier, but they don't have any chemical differences between a normal carbon and nitrogen, which doesn't have that additional neutron. And this allowed me to grow some cells with a normal lysine and other cells with this heavier lysine. And after the growth was over, I was able to combine the proteins from these cells such that all downstream experimental variation was consistent for both batches. So I was able to remove a lot of that experimental variation that may come from the different batches or from the sex replicates. And this allowed me to run the proteins on an instrument called a mass spectrometer. And a mass spectrometer looks at a mass to charge ratio for different peptide fragments. So we've taken the proteins out of the cells grown from each condition, combined them together, pulled all the proteins out and chopped them up into little peptides. And then we ran them through this instrument that let us compare peptide abundance to peptide abundance, where one of those peptides were going to have a higher mass because it was grown with a heavy lysine. And then the mass spec will further break those peptides down into secondary ions, if you will, and then backtrack to identify how much of those peptides or how much of the protein was abundant in each of the conditions. And this allowed me to assess or to quantitate how protein expression or protein abundance was different between the two conditions, or which proteins were relatively the same. And this allows us to make a statement of, if these groups of proteins that are involved in a particular process are increased, then the cell is either increasing the amount of those proteins or it's decreasing them in the other condition. And by getting a relative fold change, we can make assessments about what's happening at the protein level inside the cell. Furthermore, cells don't have brains. There's no manager or administrative unit telling every single protein where to go. So that means proteins have to be regulated. They have to know when to do something different. And one of the ways they're regulated is through a protein called SUMO. It's the small ubiquitin-like modifier. Similar to ubiquitin, which targets proteins for degradation, SUMO can target proteins for degradation, or it can prevent degradation. It can bind to a protein and turn it on or turn it off, or it can change the rate of chemistry. It can bind to a protein and change where the protein goes in the cell, it causes a translocation. Another cool thing that SUMO does is it will combine to a protein covalently. So this is an energy-dependent process where it's kind of like a strong handshake, and now you have a chemical bond. SUMO can also bind to proteins non-covalently. So you can think of this like Velcro or maybe static electricity. And it can bind non-covalently in 3 different locations. And this allows SUMO to drive these liquid-liquid phase separations, or these condensates, where the normal model is you have a cell and the cell has a membrane. And that keeps the cell's contents isolated from the other cells around it. And inside the cell, the other organelles, like the nucleus or the mitochondria or the lysosome, also have cell membranes. And that allows you to have specialized functions and specialized activities within the cell, separate of other compartments. So this process where SUMO binds to one protein and then non-covalently interacts with other proteins, and then that can interact with other proteins that are covalently bound. And the idea is that it's grouping all of these different proteins together. So why would the cell do this? Why would the cell need to bring proteins together without developing a membrane around it? And some of these processes are just for temporal cell cycles, right? The idea is the cell is going to go from step A to step B, and to do that, it needs to increase protein concentration in a localized area to drive some process forward. And then once the cell's made it to that next step, it can break those proteins down to move on to the step afterwards. You can think of like mitosis, where the cell goes through a series of steps, pulling the DNA apart and separating it to the other cells. And the idea is that SUMO creates these little microenvironments. It's almost like bringing 10 of your friends over and you're having an absolute blast, everything's peaceful, and meanwhile there's police outside at the neighbor's house, or there's a bunch of monks in one place where the city is full of violence, right? You have these unique environments taking place that are separate from the rest of the environment. And this actually plays into like cold plunges and saunas, where you don't want to break down every single protein. So when you get in the cold, sumo can help grab onto certain proteins that really matter and sequester them in, where other proteins that have bound to heavy metals or maybe lack a certain mineral or aren't functioning the way they should be or have some metabolic waste product attached to them or too much sugar. The idea is that the cell can go through and recycle those proteins and can do a process called autophagy driven by the mitochondria. And that allows you to have this cleanup step. And the same goes for a sauna where you have these heat shock proteins, or if it's cold shock proteins. And they go in and just clean things up. And then after the fact, you can undo this phase condensate like environment and release proteins back out after everything's been cleaned up. So it's almost like a safety bunker, if you will. So while it's involved in the stress response, it's also involved in just normal cellular functioning. And that's what makes sumo really unique. And it's important to realize that while DNA has an immense amount of value, it's in essence just grandma's cookbook. It's giving you the material, tells you which materials you need, and some regulation with the non-coding RNA or the small nuclear RNAs that help you say, okay, let's take these ingredients. You want to mix these things together in this bowl and these together in this bowl, and then we'll put them together later. But the proteins do all the work. Your digestive enzymes, proteins. Glucagon, GLP-1, protein. Insulin, protein. Your HDL, right? This is a high density lipoprotein, right? Proteins are really transporting, it's what's doing the work, it's what's making chemistry happen. So knowing how gene regulation changes is great, but really only 50% of gene regulation accurately accounts for what the proteins are doing or how much of that protein is present. So for this investigation, we really wanted to look at the proteins to see how the proteins respond to a simulated microgravity environment. And some of our key takeaways were that some proteins would increase or decrease, and other proteins had increased or decreased simulation, but there wasn't a direct one-to-one. Just because a protein's abundance was increased didn't mean that its simulation increased or decreased, and the same as if the protein was decreased. Which in our case makes sense because sumo doesn't regulate proteins only for degradation or only for increased activity. It has a whole slew of different regulatory mechanisms that it imparts on different proteins. So we see two variables here where the cell's responding both by increasing and decreasing protein abundance and by increasing and decreasing protein simulation in response to simulated microgravity. Now, one of the big areas that we think is a primary driver of why cells are dysregulated in space is back to this mechanosensing idea, where if gravity pushes and then that gravity tells the inside of the cell, hey, we've got pressure, then the inside of the cell can use what's called the cytoskeleton to have an infrastructure that the cell can then use to do tasks. You can think of this like the roads of a city or the I-beams inside of a building or your 2x4s, right? In essence, the cytoskeleton provides a scaffold which gives the cell structure, but then also provides a pulling force. Many of you may have seen the video where you have a polymer it's a microtubule, and you have a little dynein, this motor protein that's walking along, and each time it takes a step, burns ATP, and it's dragging this sphere, a vesicle behind it. Well, it's walking along a microtubule, part of the cytoskeleton. You also have the division of a cell. You need to have all these polymers so the cell has something to grab onto and pull the two cells apart. This also works with your immune cells, where your white blood cells and these macrophages will swallow or engulf other biological debris. And you need microtubules to create that motor force. The same with neurons and dendrites, where you've got brain cells and you've got these connective, the thin lines between. There's high amounts of microtubules in those. So you've got the microtubules and you've got actins and you've got septins and different types of polymers. Some are thicker, some are thinner. similar to our fascia and our collagen system, which some of you may be more familiar with. The idea is that our body uses these long polymers, our cytoskeleton, as a scaffold to do other functions on. So when you go into space or you're in this microgravity environment, we see that there's an abrupt dysregulation of the cytoskeleton network. Instead of having these long polymers, you begin getting fragmentations and shorter branches of all these different types of actin and microtubules throughout the cell. And it turns out that this impacts a whole host of different cellular functions. One of which was pretty surprising but exciting was DNA damage repair. The model was that when you go into space, you have radiation, and radiation is going to cause DNA damage. So we thought that was the source of the DNA damage. But in our study, and consistent with the literature, we're finding that in this simulated microgravity, or even just a microgravity environment with radiation shielding, you still have DNA damage repair proteins and genes that are increased, and you have an increased amount of DNA damage. Well, it turns out that for the cell to properly perform DNA damage repair, it needs a proper cytoskeleton scaffold to make this process happen. Turns out it also needs protein simulation. Simulation is very intimately integrated into the DNA repair process. So going into space, it's not just the radiation, it's also the dysregulated cytoskeleton that's affecting these cytoskeleton-dependent processes, such as DNA repair. Some of our most abundant protein expression changes were with DNA repair proteins. We saw nuclear porin proteins, which are the proteins involved in the nucleus of the cell that were changed. There was a lot of vesicle transport, many cytoskeleton proteins, and the mitochondria. Mitochondria has actually become one of my favorite organelles because it appears to drive the bulk of what happens in the cell. Remember, you've got DNA, which is grandma's cookbook, but the mitochondria, generate all the heat in your body. So a cold plunge forces your mitochondria to burn more fat to keep you alive. So cold plunges are helping you lose weight by preferentially burning fat. At the same time, they produce all of your chemical energy, or the bulk of your chemical energy is a form of ATP. But they also make precursors. And these precursors can be heme for your blood. They can be precursors for cholesterol, which influences all of your hormones. They make cytochromes and all these other metabolite byproducts. So in essence, the mitochondria are generating all of these different chemical precursors and heat and energy to make everything happen, but then also release compounds to drive cellular processes such as autophagy or spring cleaning, and apoptosis, which is controlled cell death. So when a cell starts to get old, or the telomeres are short, or whatever your facet of aging is, or too much cell damage, apoptosis is saying, okay, we're going to take this and we're going to throw it away. This goes to yard sale. This is antique. This we're going to turn into something else. This will set on fire. And it's a controlled manner of reusing the cell in the most effective and efficient way. And it's the mitochondria that drives that. And it turns out that the mitochondria are substantially impacted by cytoskeleton deficiencies or dysregulated structures. And this makes sense, right? If you have a power plant production facility, you need to be able to send information, send molecules out, and receive new stuff in. And if the roads or the mechanism, the cytoskeleton that you're using to do that is dysregulated, then you're not going to have an efficient mitochondria because it can't communicate properly, which is going to increase stress, increase reactive oxygen species, and increase damage. Even just simply getting glutathione in requires different cytoskeleton processes. So no amount of vitamin C or diet can help the mitochondria get to 100% efficiency if you're not building a good cytoskeleton. So in essence, there's this immense correlation with having a healthy cell with an intact cytoskeleton that not only makes you resistant to the effects of microgravity, but I think more applicable to many of you would be exercise, right? If the cell is constantly growing and shrinking these microtubules, every moment of every day it's reorganizing the cytoskeleton, then the act of exercise, the act of movement, of lifting up something heavy or even just your body weight or doing minuscule exercises throughout the day is using this mechanosensing cytoskeleton remodeling process to ensure that everything can be transported and move and function properly. So the value here really is exercise, whether it's full intensity, whether it's as long as you want to do it or as much as you don't want to do it. Every time you exercise or have really sort of any activity, you're communicating to your cells, let's be as efficient as possible. And when you take away or you become stagnant or you are in microgravity and there's no pressure, then the cytoskeleton gets dysregulated. And that means that you have increased DNA damage. You have decreased mitochondrial functioning. You have decreased communication, increased inflammatory pathways. You have decreased immune cells. So, in essence, exercise is this key function that gets the cytoskeleton and cellular communication operating properly. And that's why exercise is so valuable in preventing all these different types of diseases. And it's these sorts of insights that led me to form the LLC called Helix Prime Nutrition. And the idea is that I take these concepts, these tools that I've learned, that I've used with NASA and with the Department of Energy, to understand how the cell is functioning in order to provide insight into the molecules or processes or things you can be doing at a bulk level to give your cell ultimate health. And I've talked to a lot of doctors and nutritionists who are aware of, these different foods do these things, but if you're not looking at the health of the cell and you're not aware of how different gene regulation and epigenetics and hormonal communication, all of this plays into the cell, and how to find particular biomarkers that give you insight into the health of the cell, then it makes it really just educated guessing to say, oh, well, you should consume more of this, consume more of that in order to help the cell be healthy, help you lose weight, have more energy, reduce brain fog, sleep better, whatever it is that you're trying to do. So in essence, if you can look all the way down to the cell and you can look at the microbiome and the metabolites it's producing, then in essence, that's giving you the ultimate toolbox to make an assessment of how you go from point A to point B. And understanding that exercise, no matter what diet, no matter what food or fasting or activity level you have, exercise is non-negotiable. Because when you go into space and there's no gravity, you see advanced osteoporosis. You see drops in the immune function. You see DNA damage repair. You see rapid aging. All the things that you don't want to experience here on Earth are expedited in space. So to counteract that, we need to exercise. But the type of exercise is going to influence cytoskeleton dynamics differently. So depending on where you are, that could be a physique, that could be strongman, that could be, CrossFit. Your cells have gotten to a point where they're used to doing whatever it is they're doing. But there's a reason why they have football players and these other impact athletes do things that are very different than what they normally do. Right, ballet and Pilates and yoga. It's because one constant stimuli is not going to help the cell do everything that it needs to do. So, shameless plug, if you want to know more about how I approach cell health, feel free to reach out. I've got links in my bio. You can find, you Google my name, Jeremy Sabo, and reach out however it is you see fit. We can talk. But back to this paper, we found a few key proteins known as STU1 and STU2, right? Disco stew. We hypothesized that STU2 was going to be involved in cytoskeleton dynamics. And that's because we know STU2 is involved in nucleation of microtubules. So it binds to the very beginning kind of GABA-tubulin complex and then kickstarts the process. And then it also takes alpha beta tubulin dimers and adds them onto the growing chain of a microtubule to make it longer. STU1 is involved in binding somewhere in the middle and preventing microtubule catastrophe. So remember, the cytoskeleton is constantly growing and shrinking. It's very dynamic. So as it starts to break down, you can slap on a STU1 and it prevents that catastrophe. Furthermore, STU1 can cross-link microtubules. So you can think of this like having a string. And if you have multiple strings and you twist them together and you cross-link them, you now have a greater force. So STU2 is involved in increasing that microtubule connection. And in our data, we're showing that STU1 is decreased in protein abundance and its sumolation is increased. Now, we don't know if the sumolation is driving protein degradation or if it's an attempt to inhibit the degradation. That's a continued study. But it does make sense that if STU1 is decreased, that you're going to have these shorter microtubules because you're not preventing catastrophe. Furthermore, based on our Western blots, we have an increased amount of STU2 abundance. And this also makes sense in that if you have constantly new nucleation sites to grow this branched, kind of sporadic microtubule network, you're going to have more STU2, which initiates or is involved in the initiation of that process. So we're able to glean a few of these insights into how the cell is responding to a simulated microgravity environment at the protein level. And when we look at the literature, we see that gene expression sometimes goes up, sometimes goes down. So we don't quite have the connection between how gene regulation is regulated for these microtubule-associated proteins. But we do see at the protein abundance level that there is a definite increase of STU2 and a decrease of STU1. And that is contributing or responding to the environment of simulated microgravity. So I'm super excited about this study and this publication. The second publication that's in progress is looking at that non-covalent interaction. So this is going to tie in more of that phase separation, these kind of larger cellular networks, where in this study, it was really the very first publication looking at protein expression, particularly protein sumolation in a microgravity environment. So with that, thank you for staying tuned. And again, the takeaway, please exercise. Just do some calf raises, do some body squats, some push-ups, whatever it is you need to do. Exercise is non-negotiable. Thank you.